Hello, everyone. I'll give you an update of what I presented last year. And the objective of my project is to target metabolism in CLL. So the basic questions I try to answer is, first, do CLL lymphocytes have a favorite food? And if you don't let them have it, they will die better. Can ibrutinib change the appetite of CLL lymphocytes? Ibrutinib is a drug used to treat several hematological malignancies, but in particular CLL. And although it's a really good drug, it's not curative. Can we combine metabolic inhibitors with ibrutinib to kill them better? And in addition to that, can we identify CLL clones that will likely to respond better to some of those combinations? And when I talk about food, I'm referring to fatty acids, glucose, and glutamine, which are the food cells used to live and divide. And the inhibitors I show you just some of them here. For example, for fatty acid oxidation, a drug called etomoxir, used for cardiac conditions. Ritonavir, which is an inhibitor of glucose uptake that have been used in the treatment of HIV. DHA, which is another inhibitor of glucose metabolism, which is just alimentary supplement. A new drug in clinical testing that is used to inhibit glutamine metabolism, compound 968, and also a drug that inhibits the generation of energy by cells using all those foods. So basically, it's a bit of the first point that the previous speaker was talking, which is repurposing of drugs. Trying to see if we can use them to exploit the weaknesses of, in this case, CLL lymphocytes. And the most important, we can do this thanks to the generous donation of affected patients as the Jewish General Hospital Hematological Clinic, which over the years I have cells from more than 100 patients, for some of which at different stages of the disease. So the first question is, do CLL lymphocytes have a favorite food? And then here, okay, what you see in the y-axis is how much. In the x, in the horizontal, the food, glucose or glutamine, and each dot represents cells from a particular patient. So what we can see, and then the dot bar in the middle, it tells you is what we call the mean value, okay? So from here, what we can see is that CLL lymphocytes like much better glucose and glutamine. Like many cancer cells, they just use it in a different way. So what happened if you inhibit glucose uptake using ritonavir? So we kill them. We kill them to a different extent. So here what you see is the survival, how many cells survive. Here the drugs. And the value of 1 it means that nobody dies. If I have value of 0.9, it seems that it means that 9 out of 10 cells survive. If I have value of 0.1, it means that 1 on 10 cells die. So lower you go, better is the killing. So the killing by ritonavir of CLL lymphocytes in culture is as good as ibrutinib, the drug I was telling you before. So it seems like a good killer. In contrast, glutamine, the inhibitor of glutamine, is not as good. Okay? But what is interesting on this is that we can identify in a way which cells will respond more or less to inhibition of killing by ibrutinib by inhibition of glutamate use or glucose uptake based on some markers, known markers that are associated with the heterogeneity of CLL, including the HEVH status, mutated or unmutated, with the presence of genetic abnormalities like deletion 11q, and also with clinical stage, represented here as RAI. So, for example, in the case of ibrutinib, samples with higher RAI stage, meaning patients that are farther away down the line on the disease, will respond less to ibrutinib than samples from patients that are less advanced on the disease. And also, in yellow, you see, those are deletion 11q samples. But in vitro, deletion 11q samples and low RAI stage samples seems to respond better to ibrutinib. In the case of glutamine, you see there was not much killing if you look at all of them together, but deletion 11q are particularly sensitive to this inhibitor. So you could kill them, let's say, target them with that drug. In the case of ritonavir, it's a bit more complicated. So it's associated with, if you take the samples which have the high GVH and mutated, okay, they will be more or less sensitive to inhibition of glucose uptake, depending on their sensitivity to ibrutinib. You don't need to understand that. I don't know all the time, but what I'm trying to say is that you can, in a way, predict how the cells will die when you treat them based on things we know about them. The other question was, can ibrutinib change the appetite of CLL lymphocytes? And here what we see is the fold change in the use of glucose uptake, and glutamine uptake, okay? And also the increase in radical oxygen species that we are not discussing that today, but this tells you that, yeah, that ibrutinib can change the appetite of CLL lymphocytes because they want to eat more glucose, but much more glutamine, indeed. So what happens when you combine, let's say, ibrutinib, who increases glucose use, with ritonavir, who kill them and on top inhibit glucose uptake? So the answer is that, as I just showed you in the first slide, you get about the same killing with the two drugs, okay? When you combine them, you get a much more death with we call synergistic, because the death you get is more impressive than with each drug alone. So, of course, there is heterogeneity, and it's associated with ibrutinib sensitivity at the beginning, and the more hit are the deletion 11 again. So basically, if you treat the cells with ibrutinib and then you inhibit the use of glucose, you can kill them better. And this is another example using a different inhibitors. For example, if you inhibit mitochondrial respiration, which is CLL samples are really fond of, unlike other cancer cells that prefer to use a different mechanism called aerobic glycolysis, those cells like to use glucose in the mitochondria. So, with no surprise, that drug that inhibits mitochondrial respiration is toxic to them. But when you combine them, again, you get a big deal of death. And when you use a drug that inhibits fatty acid oxidation, that I think is one of the coolest ones, you don't get much death. The cells don't care much, except this one down here. But when you combine it with ibrutinib, they really do care. So And this is another example, I think it's the last one, basically, in which you look at how the cells die. For example, here I was talking about ritonavir induced something I told you called ROS, radical oxygen species, with many times kill cells. Oh my gosh, 1 minute. This is not, I have to change the PowerPoint. Okay. So let's say you inhibit that ROS induced by the drug, okay? And you ask yourself, okay, is ROS killing them? If ROS is killing them, I will rescue the cells. The cells will live now. If you look at the cells just like that, you say, no, there's no difference in survival. Yeah, I inhibit ROS. ROS was here, and then come down here. If you look at survival, I don't see a difference. But if you look closer to the cells, you realize that the samples that were unmutated IGVH, those guys, you rescue them. So those guys, really, ROS is killing them. But the guys that are IGVH mutated are indeed dying more. So that's what I'm trying to do, and it's complicated, and I take a lot of samples and stuff, but it's really cool. So, summarizing, what I call conclusion, therapeutic opportunities, and ongoing works. So basically, we can say that the cytotoxicity of metabolic inhibitors in CLL, which I will include ibrutinib on them, can be associated with IGVH status, deletion 11q status, and progression in the disease. All those things we know and are generally available clinically in the clinical chart of the patients. That ibrutinib increases the dependency of those cells in glucose metabolism, in fatty acid oxidation, and mitochondrial metabolism. So basically, we could combine ibrutinib with clinically available compounds targeting those pathways, and that include, for glucose, as I said before, ritonavir, which inhibits glucose uptake, and is used. It was used for HIV, but is now approved for the treatment of, I think, melanoma or myeloma. Fatty acid oxidation, as I say, etomoxir, which is used for congestive failure. And in the case of mitochondrial respiration, the best choice will be metformin, which inhibits mitochondrial respiration, but is used to treat diabetes, actually. So what I'm trying to do with my small group is expanding these studies to a larger number, because you need a larger number of patients to get statistical significance effects, and also include the worst of all in CLL, which are the lesion 17p positive samples, which have p53 not working. And this is my small lab. Gabriela, Agustin, and Sarah, and clinicians at the hospital that give me samples, Sarit and Lawrence. And again, all the patients that say yes. Thank you.